Dr. J. Bailey, and on this video we're going to talk about cell mitosis, which is a topic 1.6. So we have some of the understandings when it comes to cell division. Right over here we have mitosis, which we were going to be focusing a lot on it, looking at the 4 stages like prophase, metaphase, anaphase, and telophase. We're also going to talk a little bit about interphase, which is in the cell cycle. Now, we're also going to work on chromosomes. We're going to understand how to draw the chromosomes and how chromosomes are supercoiling during mitosis. That means they become visible. Besides that, we're also going to talk about cytokinesis, and we have to know the difference between cytokinesis in plants as well as the cytokinesis in animals. And then we're going to talk about the cell cycle, such as the phases G1, S, G2, and what's exactly happening on each one of these. And last but not least, we're going to talk about cyclins because they are the ones who control the cell cycle mostly. We're going to talk about the 4 different types and how they actually help the cell cycle move on from one stage to the next. So before we can get even to the cell mitosis, we're going to talk about 2 differences, the sexual and the asexual. Just to be clear, because some students still understand when they see the word sexual reproduction, they're talking about a sperm just coming into an egg. And that is not true. This is not what the sex is about. It's not about the sperm coming to an egg. It's about actually creating eggs and sperms. So when we say sexual reproduction, we're talking about creating egg and sperm. And this is done with meiosis. This is what meiosis is about. Meiosis is about creating eggs and sperms, and this is only happening in the ovaries and the testes of man and woman, nothing else. When it comes to asexual, in this case, we're just creating identical cells. That means usually this is when we want to repair something, like we want to replace a cell that was lost, or there's want to be growth. That means you want to go from 1 cell to become 1,000,000 cells. In this case, when it comes to asexual, we have mitosis. And mitosis is usually for eukaryotics. And we have binary fission for prokaryotics, which we're going to get more in depth. So this video is mostly focused on mitosis. We will not be talking about meiosis, but it's clear that you have to understand meiosis is just about creating eggs and sperms, while mitosis is about creating identical cells. Now, moreover, we kind of have a little bit of a scoop of how this would look in a drawing for these 2 different scenarios. So if you have asexual basically you start with 46 chromosomes, that's what the amount of chromosomes there is in humans. And this will go through the whole process of prophase, metaphase, anaphase, telophase to eventually divide into another 46 and a 46. Now, I will explain that actually before you even start the sexual reproduction, this cell already has an extra copy of 46. That's why you're able to split them into 46, 46. But then they go again through the process of prophase, metaphase, anaphase, and telophase to actually reduce to 23. And 23 is the half amount, and that's what exactly you find, 23 on a sperm or an egg, because when they join together, they will make 46. So that's the point of the sexual reproduction, is just to produce the eggs and sperms. When it comes to asexual, we start with 46, and of course we have the extra 46 because we made a copy. That will lead to 2 new cells that are identical. This will be 46 and this will be 46 because you're making exactly the copy of the parents. So that's the difference between the sexual and the asexual, is that here on this one, these guys are identical. Even the DNA is completely identical. Well, in this case, it's different. And this is a sign of a different. Why is it different? Because somewhere along the lines here, there's going to be exchange of genetic material, which will make each one of these cells completely different genetic. That's why we produce millions of sperms every day, and every single one of these sperms are different. That's why we produce millions of eggs, and all of those eggs are different. Simply because right here in meiosis, again, putting the process again for you guys to see, there's something called crossover. which you will learn in topic 3. So that's the difference between sexual and asexual. Now that we have an idea what's the difference, we're going to focus on the asexual. Before we get there, let's refresh our minds about prokaryotics DNA and eukaryotic. So prokaryotics DNA, usually they're just a circle DNA like this, and they usually are just one chromosome. There are not much numbers. While eukaryotics, on the other hand, they are usually linear and they're huge, and they can go from 2 chromosomes to a large number. Some of them can go to 102 chromosomes. So when it comes to the fact that prokaryotics, examples of prokaryotics is bacteria. So imagine bacteria only has on itself, for instance here, let's say he only has one DNA. It's simple for him because he just have to do a copy of another DNA, and then eventually what happens is it splits into 2. And now you have 2 of them. You have one here and you have here. Now this process of cell division in bacteria, we call it binary fission. And this is so easy, it's so fast because it's just one chromosome. Very simple when it comes to prokaryotics DNA, because again, it's just one DNA that is circular, and you can easily do its replication, and it's done. However, when it comes to eukaryotics, and in this case, we're talking about animals and plants, this is another story. These we're talking about 2 to maybe 102 or 160 chromosomes. It's not that simple. We have to find a way to make sure that they are organized, that we split equally, because you're not handling just one chromosome. You probably might be handling 200 chromosomes or 266, whatever is the number. Somehow you got to make sure that you are spreading the right amount to each side of the eggs. So in that process, you're going to start with one cell. But that one cell will have to go through 4 stages, which these 4 stages, we're going to focus on them on the next slides. The first one is known as prophase. The second one is known as metaphase. Then anaphase. And eventually telophase. Now, this process is known as mitosis, and that's the difference between binary fission and mitosis. Binary fission, it's very simple. It's one chromosome or one DNA and completely easily produced or replicated and then sent. However, in the other case, since we have so much chromosomes, or should I say DNA, you need a little bit of more help. You have to go through a process to make sure that it's really organized and you're splitting the chromosomes equally to each side of the cell so there is nothing messed up. So with a little bit of that, let's talk about why. Why do we want to have self-division? Of course, the most obvious thing is that these cells basically, they need to come, and they need to come from a previous cell. Now, why would you do that? So for example, imagine you have an area that you have a bunch of cells right here. And let's say that this is a tissue. So I'm going to put it here. These are a bunch of cells inside a tissue. Let's say a muscle tissue. So let's just say by chance one of these cells die. Let's say these guys gone. So when this happens, then we have an issue, right? Because there is nothing there, has been destroyed. So we have to replace the damaged or the old cells, the cells that died. When this happens, let's say this cell here or maybe this cell here will go through mitosis, and it will create new ones to repair. That means 2 is coming out of him, 2 is coming out of here. Maybe this one is going through mitosis, it produces 2, and this is going through mitosis and produces 2. So you can see, and maybe this one does this too. So you can see what it's doing. It's replacing the cells that have died. That's why it can be for repair. That's one of the reasons why you do cell division. You want to repair places where dead cells happen. For example, your skin. Your skin does cell division every day. Every time you scratch your skin, you're taking some dead cells out. And if you keep scratching every single day, every second, it becomes red. So imagine if you keep scratching. You're going to take all the cells out, and eventually going to be bony. That's why there's cell division happening every single day in your skin to replace the old cells that are not there anymore. Now, repair is one of the examples, as we can see here. Okay, so it's to replace damaged cells or old cells. But then there's also another reason why you want to do cell division. And of course, you have to know the two reasons why cell division is so important. And we just discussed that one reason is to repair, replace damaged or old cells. The second reason is for growth. What do I mean by that? Imagine you have a little sperm here and you have the egg, and they combine. Voila. And if you remember this from the beginning of the early topics on stem cells, you remember that they became a zygote. Now, that's just one cell. Imagine if we were just a zygote, we never go to cell division, we would have been just one cell. So this would have been you, and that's it, made of one cell. But we know that humans, they're not made of one cell. Okay, this guy looks ugly. I'm going to do it again. Okay. A human is made of, okay, I really know that I'm not good at this. But anyways, we are made of trillion cells. So imagine you have zillions of cells here, right? How can you go from here, from one cell, to become trillions of cells inside of your body? The only way is through mitosis, cell division. So there are two reasons why we're doing cell division. As we have said, one of them is repair, and the second one, as you can see here, is growth. These two, growth and repair, are the main reasons why we do cell division. Let's move on. So here you can see there is a parent cell, right? And this parent cell will go through mitosis, and it's very important that you understand that they are identical daughter cells. They are exactly like the previous one. So they will have exactly the same DNA and exactly the same DNA as the parents. That's what mitosis is about. It's just making identical daughter cells. Now, when we talk about keeping cells identical, we're talking about keeping the DNA identical. So let's just say on the first cell here, let's say that this is the DNA. And you want to make new cells. Let's say this is the new cells that you want to do. In order for you to be able to get these two new cells that are identical to the parent, you have to make a second copy that is exactly the same as this one. So they have to be exactly the same. When you make it exactly the same, now that you have two copies that are identical, right? Then you can give one of these copies to the first cell right here, and you can give the second one to the copy right here. And that's what's about doing cells that are identical. So if you notice from this, you cannot start cell division without making the copies of the DNA first. It is extremely important that the first step you do is replicate this DNA here. That means make a copy first. Once you do the copy, then you can go and do the cell division. So, before we get into cell division, let's understand more about chromosomes and DNA, because some students still don't understand the difference between this. Now, we know that DNA is what carries the genetic informations, right? And we carry this stuff inside a chromosome. Now, we have many different types of eukaryotics. We're talking about small little plants to big animals. Some have 10 chromosomes, some have 50, some have 92. Like we have 46. We have 46 chromosomes, and 23 of them are identical pairs. And we're going to explain a little bit more about this. So you have a picture right here that it shows you clearly what the chromosome looks when it's actually visible. We're going to understand a little bit more of that. But before we do, let's understand what it means. Right here, this image that you can see here is basically a bunch of DNA, but it's not visible. And what I mean by not visible is that you cannot tell me here, if you can figure out how many lines of DNA, how many lines of all these genetic information is, or how many chromosomes there is, I probably will be some kind of Superman, because it's impossible. I have no idea how many there is. So in this case, they are uncondensed and invisible, uncoiled. You just cannot see them distinctly. It means you cannot count the amount of chromosomes there is, simply because they are uncondensed, they are invisible, and they are uncoiled. Now, we call this chromatin. So chromatin is just a mixture of a bunch of DNA all around, exposed without any coiling. But then we can actually coil these guys, or should I say organize them, and then they go from this to become this. When they become this, now you can see a chromosome visible. This has actually been condensed, it has become visible, and it has become coiled. Now, it's difficult to understand this, so I will definitely draw and show you how we go from being something that is completely uncoiled to something that is coiled. But you clearly can see that here, there is no chance to count, but here you can count. That is one chromosome. And obviously this one is easy to see, while this one it's impossible to see how many there is. So right here, let me show you what happens. So let's just say you have a DNA like this, and you have another one here, and you have another one here. And if I keep doing this, so many different DNAs here are lining up. And then I ask you, okay, how many have I done? You can stop the video and go slow and see how many times I've done. But here you can see that is completely invisible. You do not know how many chromosomes how many DNA I have actually drew here. But how can I make this guy become visible? Now, if I drew this, if I do like this, now you know how many I have here. I have, let's see, 3, 6. There's 7 chromosomes. So what this means, this was actually visible. So I want to get basically from invisible to visible, because visible I can count, I can see them. Now, what I gotta do, I have to condense it. Condense. And condense is the same thing as saying that I want to coil them. How would I coil them? So think of a line here. You just have a line. And then imagine I have a ball here. So what I do with the ball and the line, I will take this line right here and I will wrap it around it like this. And my wrapping around, now, what does it look? You have the first ball here, and I have it lining like this. I have the first chromosome. Let's say I have another one. And I can take the second line of DNA and cross it. So now look, I know there's 2. Why? Because they have been wrapped around something, now they're visible. And I can do zillions of that. So I can take each one of these lines that was here, and each one wrap around this one little circle. Now, what is this little circle that I keep talking about? It's a protein. It's a protein ball that has a specific name. It is known as histone. So DNA is wrapped around the histone, and when the DNA is wrapped around the histone, it becomes visible. So how would the chromosome look like? Well, let me give you space here so I can show you what I'm talking about. Let me make the bigger one here. What I'm trying to say is this, and you're probably gonna know this. If we take one DNA, here is one of a protein histone, and I take another one, and I take another one, and I'm just using protein, or should I say histones, right? And I'm building a copy of it. Actually, let's not do this one for now. So I have this. Now I will take the DNA, which is long, and I will just wrap it around it. Wrap around it. This is one DNA, just one, and it's wrapped around these histones, right? Now, if you notice, if I put a line here, what did this show? This is the same thing as saying one chromosome. And if I want to make a copy of this chromosome, I put it again, another one here, another one here, another one here, and then I wrap the DNA, a copy of that DNA again on around it, and look at it. I have made a copy of each other, and now I am attaching them together. Let me put another color here, by this thing called centromere. What do we have here? We literally has a chromosome. So what does this shape here looks like? This shape looks like those little X that you find in the pictures when the chromosomes are visible. It's simply visible because look, if you look inside, they have been coiled around a histone, a protein. So if you do all the DNA wrapped around these histones, then eventually all the chromosomes in the cell will become visible and they will look like X's. So this is how you actually make the chromosomes visible. Just to make it more clear, now you can have DNAs all over here, bunch of DNAs, and again, these are invisible, they are uncoiled, and that means they're uncondensed. These happen during the day when they're not dividing. They're basically impossible to be seen. But once you coil them by wrapping them around a histone, what do these guys turn? Suddenly they are going to start turning into like this. Why? Because they have been wrapped around the histones and now it's more clear. So what happened? They have been coiled and now they are condensed, and this only happens during mitosis because you need to make sure you can see them in order to divide them. It's the same thing of thinking, imagine this is a spaghetti. There's no way that you can look in the spaghetti pan that you will know how many spaghetti lines there is there. But if you separate them in two pieces with each other and wrap around, you will be able to see the difference. So basically it's the same kind of logic. So here is more like a picture of a drawing that you can see. And you can notice that the red thing here, this is actually the histone, and you can see that the blue one here is the DNA. So if you can see, the DNA is inside and it's wrapped around these histones. These are very important, histones, and there's so many different types of histones, but you don't have to know them all. You just have to know that there will be a DNA wrapped around it, and there's a clip of histone that is making sure they stay there. So this picture probably will make it much clearer for you. So what you see inside here, you can see that the DNA has been wrapped, coiled, and it's been coiled by the histones, and it forms the shape of an X that you can see. And you can notice that right here in the middle, that's where the centromere is. And then there's always going to be a short arm, and there's always going to be after the centromere a long arm. Now each one of these guys is a chromatid. So basically what are we saying? We're basically saying that this one by itself is a chromatid and this is a chromatid. Together they're known as sister chromatids. Why do I say sister? Sister means because they're the same. The DNA of this guy is exactly the DNA of this guy. It's a copy of each other. And the copy always stays together by the centromere. It holds the two copy exactly together. Now, we might have a little bit of confusion with what we call sister chromatids or chromosomes, but chromatid is eventually a chromosome, not a chromatin. So you got to be very careful with that. So now that we have seen a visible chromosome and understanding more about it, let's just be going through the mitosis. Actually, let me just show you one thing to make it more clear before I start with the mitosis about the difference between chromatin, chromatid, and chromosome. Because these are three different things. Look, chromatin, and then there is the chromatid, and then there is the chromosome. And these interchange names, but see, chromatin, this is invisible. You cannot see it, so it's like that the smash of a bunch of DNA like this. Chromatin is just made of DNA that is invisible, and you cannot see much. And usually you can have some proteins around it, but they are not wrapping around each other, so it's just invisible DNA. Chromatid, in this case, now the DNA, I'm going to put here again the histones, and then I'm going to have the DNA wrap around it. So you can see that this right here is a chromatid. If I want to make a copy of it, then I do it again, the histones here, and I made a copy, wrap around it, and voila, I have its sister now. And the sister, all those two are bound by a centromere. So in this case, one of this is a chromatid, and the other one is also a chromatid. Now, together, they're known as sister chromatids. And they're, again, like I said, sister because they're identical to each other. So then the issue with students comes, well, then what the heck is a chromosome? Believe me, it depends on what the situation is, but guess what? If it's just this one by itself, it can be called a chromosome. And when they're together like this, it can also be known as a chromosome. It's interchanging throughout the entire process of whatever IB you're learning. But once we get to the stages of mitosis, I'm going to show you how you can easily talk about it. But chromosome is the same thing as a chromatid. It just means it's wrapped around the histone. Okay, but when you have a sister with it, we don't talk chromosome, we just say sister chromatids. When it's a chromosome on its own, like this, if it's a chromatid on its own, we can call it chromatid and we can call it chromosome interchange. But when we put his sister, then we avoid saying chromosome, we just say sister chromatids. Hope that helps. So with that, let's start mitosis, which is an asexual reproduction. So the first thing we have to know is the 4 stages of mitosis. We have prophase, let's get some color here. We have prophase, which is the first stage. We have metaphase, the second stage, anaphase, and telophase. Right here, you have 4 images that are found in the microscope, which, by the way, you really have to be really good in spotting them, because you might get one of these questions in the multiple choice where they put one of these 4 pictures and tell you, hey, is this a prophase or is this a metaphase? So starting with prophase, this right here, number 1, this is the prophase. The chromosomes are becoming visible, as you can see. Then metaphase, if you look at the word meta is the same thing, closer to the same the word middle. So where do they find the middle? Oh, here, they found in the middle here. So that would be number 2. Then anaphase, the word ana comes from anarchy. And what happens in anarchy? Anarchy, we start to separate, everything is chaotic. So they're going to each other. Oh, some students usually use anarchy with the word a as it means going away. So you can see there some chromosomes are going away and some of these are going to the other side. So this is the 3rd stage, the anaphase. And telo, because telo is the same thing as a tail, and a tail is the end of something. So we can see at this stage, you're already starting to see 2 cells being forming, and you can see the chromosomes are becoming invisible again. So this case will be number 4, the telophase. So these are the 4 stages of mitosis. Now I'm going to go through each one a little bit more detail and what are the things that you need to know to describe them, as well as to draw them. So starting with the early prophase. If you remember, I mentioned about chromatin. That chromatin, remember, it's a bunch of DNA like this, and you cannot see, it's invisible, right? What happens? They, inside the nucleus, they start to condense. When I say the word condense, that means they are starting to be wrapped around that protein, the histone. So now they have become visible chromosomes. At the same time, you have something called centrioles. And the centrioles are those little stars that basically have spindles coming out of it. And when the spindles coming out of it, these spindles will be very important to the separation of chromosomes. So some of the things that you really need to talk about prophase. First, you have to mention that the chromosomes are becoming visible. That's the most important thing. Then you have to mention the centrioles are starting to move to the opposite side of the poles. That means one is coming to the side, the other one is coming to the side, and you can see the spindles are starting to come out. That's very important. On the late prophase, you can see that the chromosomes are becoming extremely visible now. And you can see that the nucleus envelope here is breaking down. That means the nucleus wall is breaking down. So when we talk the entire prophase, you can say that the nuclear membrane and nucleus is kind of broken down. So that means the wall of the nucleus is broken down. You can say the chromosomes are becoming visible, they're condensing. You can talk about the spindle fibers, they're coming out. And the kinetic core, it basically means a part of the spindle that is attaching to the chromosome, right, specifically at the centromere. And you can see that the poles of the cells, you will have the spindle forming eventually. So this is what it would look like if they told you to draw a prophase. So in prophase, to make your life easy, the first thing you would do is make sure you draw a solid line to be the cell, and then a dotted line. You have to draw the dotted line because the dotted line means what? This. First thing I would say is the nuclear envelope. And by the way, the word nuclear envelope means the wall of the nucleus is breaking down. The next thing you're going to say is the chromosomes, they become visible. So you're going to have to draw them visible. That means you should be able to count them. So number 2, chromosome. Or you can say chromosome becomes visible or chromosomes become visible. Either way, it's okay. Oops. Let's erase it and put this back in. So the chromosomes become visible. And then let's talk about the centrioles. Let's use the orange here. So the centrioles, this is a centriole. They usually have 2. So each one is moving to the opposite side. So that means to the poles. Oh. So the opposite. They're moving to the opposite sides. So number 3, you can say the centrioles are moving to the opposite side. So again, centrioles are moving to the opposite sides or the opposite poles. Now, as the centrioles are moving there, right, as they're moving, spindles are starting to come out. So you can say number 4, spindles are forming, and you will get the points for this. So this is the drawing of prophase. You don't have to do the other organelles and anything, but if you had to ever write the stages of prophase, which is the first phase, first, make sure you mention that the nuclear envelope is breaking down. Second, mention that the chromosomes are becoming visible. Third, mention that the centrioles are moving to the opposite side. And the last thing, make sure that you're saying that the spindles are forming. They're coming out of the centrioles. Now, having those 4 descriptions, you nail the prophase. And again, make sure the drawing is clear. For instance, what I mean by the drawing, make sure that this nuclear envelope is being dotted lines. Because if you make solid, it doesn't show that it's breaking down. So make sure the nuclear envelope is in dotted. Also, make sure the chromosomes are visible. Because if you make it all like a smash a bunch of spaghetti together, that means you not prove the point. Make sure they're visible. And also make sure that you have a line that shows that they're moving to the opposite poles. And you can have some of the spindle coming out like this, or you can have the spindles already coming out as they're moving like this. Have them already come out like this is a sign. So this is prophase for my mitosis. Now let's move on to the next stage. Oh, but before we do, I forgot this part. So if you can recognize here, as you notice, this right here is a chromosome, right? This is a chromosome. And you can see right in the middle here, what is this? This is the centromere. Now, what's the kinetic core fiber here? This is part of the spindle. So this yellow thing is actually the spindle. And the spindle, what is it going to do? The spindle is going to attach to the chromosome, which is this purple thing that we have here. So the spindle is going to attach right here at the centromere. That's the purpose of the spindle, is to attach to the chromosomes. Now, where exactly does the spindle attach? Right at the centromere of that. So wrapping up prophase, the things that we see. Look, you have to know how to identify it in a microscope. So what you have here is you see this distinct blues. It's starting to become visible. And if it's becoming visible, that means it's actually going through prophase. Like here, for instance, they're becoming visible, as you can see, and eventually they're going to reach a point where you can see them completely. Also, look at this. Right here, you're seeing the spindles and the centrioles. And you see the spindles coming out of the centrioles as they move into the opposite, because in the end, they want to be the outside. Moreover, you can see here that the kinetic core, basically, which is part of the spindle, is finally attaching to the centromere of those chromosomes. So here in the bottom, you have everything that I mentioned, how the nuclear membranes is being disintegrated, that means broken down. The nucleus disappears. The chromosomes are condensing, which that means become visible. The spindles, they're beginning to form. And then the kinetochore, which is part of the spindle, it's actually attaching. Now, this is actually attaching to the chromosome, not spindle. So this part right here, you should be saying chromosome. It's attaching to the centromere of the chromosomes. So this is a part of the prophase. Let's move to the next stage, which is metaphase. When we match the metaphase, like I said, the first thing right here, this, it means to the middle. So when we say middle, that means the chromosomes are lined up right in the middle. And when we say middle, we can say the word equator of the cell. Every time I say the equator, equator is the same thing as the middle. So the sentence right here where it says chromosomes are now lined up in the equator, you can say also the chromosomes are lined in the middle of the cell. At the same time, who actually took these chromosomes to the middle? It was actually these spindles right here. These spindles here attached the chromosomes and pushed these chromosomes to the middle. That was the purpose of the spindle, is to put these chromosomes in the middle. And that's metaphase. It was just making sure that all the chromosomes are attached to the spindle and moved to the middle. So here's an example of it. You can see it right here that you have the centriole, right, on the two sides of the cell, and you have it right here in the middle, the chromosomes line up, and you can see that the kinetochore, right, this little thing here, which is part of the spindle, is attached to where exactly? The centromere. Where is the centromere? This is the centromere, centromere, centromere. So that means the kinetochore of the spindle is attached to the centromere of these chromosomes. And these chromosomes, they did not just move randomly down there. It was the spindles here that touched them and pushed them, all of them, into the middle. That was the purpose of metaphase. So if I had to draw, if the teacher asked you to do metaphase, how would I draw? First, make sure you do a line of a plasma membrane circle. Make sure you have the centrioles on the corner. Make sure you have chromosomes line up. Now, if you notice, I drew on the previous one, if you look back here, all the way back here when I was drawing, I drew 1, 2, 3, 4. Since I drew 4 chromosomes, make sure that you're always doing 4 because they like you to see that you're always using the same numbers. And then I have the spindle that is coming out of the centromeres, and they are attaching to who? The centromere of the chromosomes, or the sister chromatids in this case. So you can see that they are attached to this. And where are they located at? All of them are located right here in the middle. That was the purpose. So metaphase, it's a very short description. You can say that the spindles fully form and attach to the centromere of, you can see sister chromatids or chromosomes are now in the middle of the cell. So it's just one simple description. The spindles are attached to the chromosomes. Where exactly? At the centromere. And they are basically lined up in the middle. You also can say that the chromosomes now are lined up in the middle. And you will get points by just saying that the chromosomes are lined up in the middle as they are attached to the spindles. And that will give you the full points for metaphase. So here's a picture in the microscope. You always try to spot here basically the spindles here, and you have basically, when I say aster, that's the centrioles, and you have the spindles here, and the blue here is basically all the chromosome, and you can see the chromosome is right here, line up at the equator, the middle of the cell. So it's very easy to spot them in a microscope when you see them completely in the middle while you have the red on the sides. And this is real vision, so always the red stuff will be basically the spindle, and whenever you see the blue has been highlighted to be the chromosomes vision. So in the review of metaphase, we already saw the picture on the microscope. You can also see in the drawing that they are lined up in the middle just as they are lined up here. And you can see that the chromosomes are attached to the kinetic core of the spindle, and they're basically lined up where at the equator of the cell. Just mention spindle being attached to the chromosomes, and they're lined up in the middle of the cell or in the equator of the cell, and you got the points from metaphase. That's a very simple stage. Let's go for the next stage. So after metaphase, anaphase come. This one goes really fast. So if you remember, it was attached, there was two, right? Say it was sisters. So now the sisters have been pulled apart. So you have one of them going this way and you have the other one going the other way. And you see the other one going the other way. So they're being split. Those two sisters, or those two chromatids that were exactly identical, now each one is going to the opposite side. So they have been pulled by the kinetic core. They have been pulled by the spindle. And what happened is the spindle is actually moving in this direction. So it kind of split them away from each other. And they're separating. One is going to one side, the other one's going to the side. And why they're doing this? Because in the end, they want to have an equal amount on one side and you want to have an equal amount on the other side. Because remember, it's all about making copies. So that's what anaphase is. Anaphase is just this simple sentence here. The sisters are being pulled apart to the opposite side of the cell. That's it. It's just being pulled by the spindles to go to the opposite side. That's all it is. So if I had to actually draw the anaphase in a test, how would I write the draw anaphase? First, I will have continue to be a circle. I will have the centrioles right here. Now I make the spindle a little bit shorter. And remember, I'm only doing 4 because I only had 4. So I'll have the first chromosome, or should I say one of the sisters, another sister here. So you can see that before they were like this. And what happened is I pull one way and pull the other one the other way. So I split the sisters. One sister is going to one side, the other sister is going the other side. And that's the drawing of anaphase. You don't have to do anything else. So in that one sentence, I will just say the spindle split the sister chromatid as each one move to the opposite side of the pole, and that's it. Oops, pole. Can't write pole anymore. Look at that. Or just move, each one is moving to the edge of the other one. That's pretty much it. It's just one sentence. Sister chromatids are being split and they're moving to the opposite sides or to the edges of each side of the cell. And that will be your points for anaphase. So here's what it looks like in a microscope. Again, you have to see it. You can notice that there is a motion going on. Now, some people are going to say, no, but that looks like two cells. No, if you really look closely, let's look at it here. If you really look closely, right here in the middle, you don't see a black line like you see a black line here. You know, you don't see a black line. You don't see a black line. So there is none here. So you can easily know that it's still made of the same cell. So because there's no line, you know that this is one sisters are going one side and the other sisters are going to the other side. They're being separated. It's not like they have that black line that is separated, because all of this is that one plant cell. That's how you can spot that the anaphase is actually separating. So reviewing it again, this is what it looks like. You can see that they have been split right here, right? You have the half of them coming this side and you have the other half here. And you have the centrioles and you have the spindles becoming smaller. So it's anaphase, it's all about separating the sister chromatids. And these sisters, each one are going to each side of the pole. Then we come to the last part, the last stage of mitosis, which is telophase. Now, telophase is a stage where the sisters have finally reached the edge on each side, and there's no more spindles, right? So they're already on the opposite sides. Let me use another color. They're on the other sides. The spindles, you don't need the spindles anymore because the spindle's job was just to separate them. Now you have the nuclear envelope coming back, and that nuclear envelope is coming back around these sisters. And that means the nucleus is appearing, which is the location of all the DNA. And now the chromosomes are going back to being chromatin. That means they're being what? Uncondensed again. They are uncondensed and they go back to being invisible. That's what chromatin was. If you remember, chromatin in the beginning was just DNA all around. You couldn't see it. So telophase is the final stage of mitosis, in which everything, but there hasn't been division yet. That's the most important thing. We reach the stage where the chromatids are on each side, and you have the nuclear envelope coming back, and the chromosomes become invisible, and that's where telophase stops. But if I had to draw this in a test, this is where students might get points off, because they do this. They make two cells, and then they make this, and they make this, and they draw this, and they draw that, and they make the centrioles here. If you do this, you lost your points. Here's what they really want you to do. That's not telophase. This is not how telophase looks. Telophase looks like this. They are still connected, but it's starting in the middle to be split. And then you have the nuclear envelope come back. So now you make a solid line for the nucleus, and you make a solid line for the nucleus on the other one. And then DNA is invisible. So you just see lines up like this. There's no more these visible X's anymore. They're not there anymore. They become invisible. And you can see that the line right here, what happened, it's starting to bend. That means the cytoplasm is starting to cut in the middle. But that's it. That's the drawing of a telophase. And so if I had to write what telophase do, you can say chromosomes become invisible. You can use the word invisible, or you can use the word uncondense, or you can say become uncoiled again. Then 2, you can say cytoplasm starts to split. Not yet. And number 3, you can say that the nuclear envelope, let me erase this part so I give space. The nuclear envelopes reappears. So you have the nuclear envelopes coming back around these guys, as you can see here. The nuclear envelope comes back around, and you can see that the chromatin, not chromatid, chromatin is back there, where it's just DNA that is not visible. That's telophase, and that's how would you get the points for telophase. So you even have here our comparison of anaphase and telophase. You can see just from the blue here is on the corner. Here they're still moving, so you still can see a little bit of red here. Also, you can see here is empty, much, not that dark white like this one that you see here. So you can easily see the difference between an anaphase and telophase and not get confused on it. You can see from the drawing too, right, that here they're starting to build up. They're packing together, right? They're packing on the corner, they're packing in the corner. However, here doesn't show the circle of a nuclear envelope, so I would put it. And here you can see they're splitting. And here they're still a little bit visible, so they should be like this, invisible and stuff like that. So anaphase, it's all about separation of chromosomes. Telophase, the chromosomes become invisible. So you can see that the nuclear membrane eventually will start to form here, and another nuclear membrane will start to form here. And these chromosomes will not be able to see the difference between them. They'll be really dark like that. So that's the difference between anaphase and telophase. So wrap it up. If we want to go through the whole process of mitosis, as we have seen, mitosis right here has prophase, then it has metaphase, then third it has anaphase, and the fourth it has telophase. And the whole purpose is just to separate. Oh, my handwriting is going crazy now. Separate chromosomes equally. Now, what I mean by that, just to be clear, when you are going to start mitosis, you are starting with a certain number. So for example, we know that humans are made of 46 chromosomes. But if I want to make a copy of this, then first I'm going to make sure that I make the extra 46. So I'll make the extra 46. So what does that mean? Now I have 92. Then I will split them into 2 in the end, which is going to be 46 and 46. Now, if you remember from telophase, we didn't finally split. So there is one thing that happens after mitosis, which is cytokinesis. And we're going to get to them soon, right here, which is cytokinesis. It basically means the division of the cytoplasm. That means you finally cutting the cytoplasm in half. And this is when you finally get the 2 daughters. So you do not get the final 2 daughters in telophase. You get the final 2 daughters. And what I mean by 2 daughters, I mean these 2 daughters here, only after cytokinesis. The word cyto here means cytoplasm, and kinesis means the movement of the cytoplasm. Now, in plants, we call it cell plates, and animals, we call it the cleavage furrow. So I'm going to explain a little bit more about these guys right here on the picture. So cleavage furrow is, as you can see, this is the telophase. Look what happens. It's starting to squeeze right here, and it's causing a cleavage. And that cleavage is breaking out, and eventually it's going to split into 2. Cell plate, usually they form a wall, and by using sugar. And I'm going to explain a little bit more how that works. So you have to know the difference between cytokinesis in a plant, and you have to know what cytokinesis is in an animal cell. And they have their specific name. So it's extremely important that you know that cleavage furrow, it is the name we give for cytokinesis in animal, and cell plate is the name that we give for cytokinesis in plant cells. Before we do that, let me go back here just to make it clear for you to see what I mean by that. That maybe this will help you be able to understand cleavage furrow as well as cell plate. So first, let's talk about the cleavage furrow. This one is actually happening in animals. So how does it work? If you remember in telophase, it was like this. And you had the invisible DNA with the nuclear envelope on each side, right? All done. So this is telophase's end. Now, you see right here, and I'm going to highlight this part right here, that you need to finish closing that gap. And that means you have to split eventually the cytoplasm right in the middle. Now, I usually tell my students in school that this looks like a woman with her cleavage, and that makes you remember very easily. I know some teachers in some schools will kill me, but since I'm not in school anymore, I can show you this. And you can see cleavage. There is cleavage going on, and furrow is just going through the middle, which you knew Latin, as in perforating the middle. So that's what looks like an animal. You just start squeezing from the middle, and you squeeze until it eventually cuts in the middle. That's what it does. Now, moving on to this, now let's look how it looks on plants. How does a cytoplasm split in plants? So first in plants, we call it cell plate. That's the name. You need to know it. And like I said, this is a cytokinesis in plants. So here is a plant cell. Now, what happens is plants do not split equally. That's why sometimes one branch is bigger than the other side of the branch of the tree, is because they don't care. So you might have some chromosomes here, and then you might have some here. That what happened is you have these little granules or vesicles. And what do they have? They have sugar. In this case, it can be cellulose and starch. Now, what do? They all start moving into direction of one area. And when they get here, let's pretend that these little dots inside here are sugar, right? So when they get here, they start releasing their sugar. And they keep releasing their sugar until this entire sugar layer comes all the way to the 2 sides. And then eventually they start to form a wall. And that's how you get the cell wall. Voila! For a plant. Now here's the interesting part. You can see that this is smaller and this is larger. So with plants, there's no such thing as cutting these plants into 2 cells that will have the same size like in animals. In plants, sometimes one cell will come out very small while the other ones come very big. And that totally makes sense why when we have trees, some branches are really big and some are tiny. It's just that when it comes to plants, we don't split the cells equally in size. Some can be big, some can be small. However, remember, in animals, they split equally. They come out with the same size, with the same DNA. In plants, you do have the same DNA, but they don't come out with the same size. So this is an example of cell plates. You just have a sugar granules releasing right in the middle and causing that cell plate, which that cell plate eventually becomes the cell wall. That's the cytokinesis for plants. So now we got the cytokinesis. Since we already talked about mitosis, prophase, metaphase, anaphase, telophase, which is basically the cell division in animals and plants, now let's talk about the cell and how does it live. We talk about his cycle. So when it comes to the cell cycle, there's 5 phases. We already went through the mitosis and cytokinesis. We described those two. But this is when the cell wants to actually divide, right? When it wants to divide. Now, when the cell does not want to divide, what does the cell do while it's not going through mitosis? Well, it has 3 stages. It has G1. And G1 is where he's just growing, he's living his life, viva la vida loca, as we say in Spanish. We're just having so much fun with it, and they'll continue doing whatever. So if it's a heart, it will pump. If it was a lung, it's breathing. If it's a cell in your muscle, then it's running. If it's your tongue's muscles, it's just talking, whatever it is. The cells are just living their day-to-day living. However, if the cell starts feeling, hey, dude, I want to start dividing. So basically what it says, well, if you want to divide, then you have to prepare yourself for it. So what would you do first? I'm going to make a copy of my DNA, because if I want to split it to 2 cells, I got to make sure I have 2 DNAs that are identical, and I can give to each cell my DNA, identical. So the S stage, which we know as the synthesis, which means to make, we're making an extra DNA that is the same copy. So it's basically DNA being replicated, or we say DNA replication. Then we have G2. This is the secondary growth phase. This is the time where you make extra organelles and you give more cytoplasm, and this is when the cell is just coming bigger and bigger because it's about to split. So these are the 5 stages of the cell cycle. Now here is a picture of a drawing that we can actually do this in a test. So in this cycle, you can see that it starts with G1, right? This is where they're growing and living their metabolic lives. And then you have the S stage. This is where DNA replication. This is when you're making an extra copy of DNA. And then you have G2, which they continue to grow and produce more organelles. Once they reach here, this area right here, this is the moment they say, hey, I am ready to divide. I have my extra copy of DNA. I have grown to the limit I can grow as a size. Now I want to divide. Well, since you want to divide, then you have to go through the process of mitosis, because mitosis makes sure that you divide equally. That means each cell will get equally the same amounts. So for example, if a cell is made of 46, you want to make sure the new cells are made of 46 too. So with that, we go through prophase, metaphase, anaphase, telophase, and then you have cytokinesis to split the 2 cells. Then voila, you have 2 new cells. What does those 2 new cells do? Both of them enter G1 and grow in size and live their lives and live their functions until the moment they feel they want to divide again. Then they enter the S stage here, and then they make a copy, and then G2, they grow more, make copies of their organelles, and then boom, they're back again into mitosis. And that's the cycle of a cell. It keeps living its life. Now, the interesting thing about this is that some cells never, ever leave G1. They live forever in G1, like the brain cells. These guys, they only stay on G1. They do not go to mitosis. Now, we also know that most of the cells live most of their lives in G1. The S and G2 and the rest of mitosis, this is just a temporary thing of 24 to 48 hours. But G1, they can live 20, 30, 40 years on G1 stage. So, recapping here, let's talk about G1. So G1, of course, is the first stage. Here the cells are maturing, the organelles, remember, because they came out as baby cells. So they're growing and the organelles are doing their functions. They continue their metabolic activities. When I say metabolic activities, I'm saying, for example, imagine you did a heart cell. What will be the metabolic activity of a heart? Well, the heart pumps. or making blood out of it, pumping around, whatever it is. So they're just living their lives. And then, as I said, they spend most of their life at G1. So the heart cell will stay most of 70, 80 years just pumping blood. That's G1. S stage, which the word S comes from the word synthesis, which means making. You're making something. What are you doing? You copy DNA, or you can say replicate. So here is the original DNA, and what are you doing? You're making the extra 2 copies. They are both identical. It has to be identical because, again, mitosis is about making copies. Once you finish copying, then you move to the next stage, which is the G2. What the G2? That stage of growth, you have DNA is already then copied, right? So what do you do next? You just continue building up the next things, like the centriole, the mitochondria, which is the organelles. Some proteins are built up. So, like, for example, you want to make more histones because you want to make sure the mitosis has the chromosomes visible. So everything extra from the organelles, cytoplasm, they all being extra copied here in G2 phase, as well as the cells getting bigger. Once this is done, we get back to mitosis, and I'm not going to go through the process of mitosis because by now you know. If you don't, go back to the video. With that in mind, if the teacher asks you to draw the cell cycle, that's very simple. You just write G1, an arrow, S, an arrow, G2, an arrow, mitosis, an arrow, cytokinesis, and then go back to G1. More detail. Then you write growth and development. Here you can say synthesis, or some, even IB will allow you to just say the word DNA copy. And then here you can say growth and prepare for mitosis. Or you can also say making extra organelles and cells getting bigger. Then here I would put the 3 out, writing prophase, metaphase, anaphase, and telophase. And you can write here mitosis, the whole word. Don't just put M because some IP do not accept one letter. You have to put the whole thing, mitosis. And then cytokinesis, you just say the cytoplasm splits. That's it. And then the cells, the new 2 cells are back to G1. There you go. Now, here's one thing that I have not mentioned on the previous slides that is very important. When we come to G1, S, and G2, this stage, we call it the interphase. Interphase is the stage where there is no mitosis, where there is no cell division happening. Interphase is basically the stage where there's growth, there is DNA replication, there is organelles extra, there's the living of the cells. That's what interphase is all about. So interphase is made of G1, interphase is also made of S, and the interphase is made of G2. Interphase is made of these 3 sections. However, mitosis and cyclins are not part of the interphase. They're outside of the interphase. Make sure you know this well. So now that we have talked about the cell cycle and we have talked about mitosis and its stages, now we need to know how are they actually regulated. When it comes to regulating the cycle, we're talking about this. We're like, how do you know when it's time to move from G1 to S? And how do you know it's time from S to go to G2? I mean, yes, you make the DNA, you're finished doing the DNA, but then somebody has to communicate, say, hey, dude, we have finished making a copy of DNA. So what do we do now? Oh, there will be a signal that says, okay, buddy, I guess it's time to go to G2. When you get to G2, when do you really know it is time to enter mitosis? So there is a specific type of protein that helps regulate the cell cycle and tells it. This protein is known as cyclin. Cyclin is the one that regulates the cell cycle. Now, the cyclin doesn't work alone. There are enzymes that are called cyclin-dependent kinase, which we know as a CDK. So you can see the word dependent. It depends on the cyclin, and kinase is a way of an enzyme that basically is working on doing phosphorylation. But you don't need to know that. You can work even with the word CDK and you're fine. So it's cycling and CDK is the enzyme. So we have the enzyme and the cyclin. Cyclin is the protein, enzyme is also protein, but cyclin and CDK, these two together, they will regulate. So what exactly does the cyclin do? Cyclin actually activates this enzyme. So the cyclin here actually activates CDK. And every time cyclin activates CDK, you go to the next stage. But for cyclin right here to activate the CDK, they have to have a high concentration. So when they have a large amount of cyclin, then CDK is activated, and that means we move to the next stage. However, after they move to the next stage, cyclin drops again its amount. And you just keep doing this. So the cyclin go back to being low. Now, there's 4 of them, and we're going to talk about it right here. There's 4 types of cyclin. There's so many different types of cyclin, but you only need to know 4, okay? These cyclins are cyclin D, cyclin E, cyclin A, and cyclin B. So these are the 4 cyclins. So there's 4 cyclins that we're working with. As you can see here, we have E, A, B, and D. These are the 4 cyclins that regulates. And the 4 of them, they actually work on who? On CDK. Now, CDK can only be activated with large amount of cyclin. Oops, wrong writing. Exactly. So CDK can only be, it's an enzyme that can only be awakened or activated if and only if you have a large amount of cyclin. Now, each of these cyclins has a purpose on the stage. As you can see here in the drawings, you can probably see that if you want to go, let's put another color here. Which color do I want to go with? Let's go green. If I am in mitosis and I want to move towards G1, I need to have a large production of cyclin D in order to activate CDK to go to G1. If I want to go from G1 to S, what's the cyclin that I need? E. I have to have a large amount of E to activate CDK to move to S. If I want to go from S to G2, I need to have a large amount of cyclin A to move to G2. And if I want to go from G2 to M, I have to have a large amount of cyclin B. That's what these drawings in this diagram is basically showing. So if I want to wrap this in a nice, easy way for you to remember all this, this is how I do it. Let's start with mitosis. M. Okay, let's use actually black. So let's put this out. So if I want to go from M, if I want to go from mitosis to G1, what do I need? D. If I want to go from G1 to S, I need E. If I want to go from S to G2, I need A. And then if I want to go from G2 to M, I need B. So it come out as Deb. Well, not Dad, but Deb. That's the one. Let me put it here. It looks ugly in my handwriting. Let's say here. So Deb. And this is how you get back to the stage. So how can you memorize this? This is the little cheat sheet that you can have in a small piece and write it down, and it will come out perfectly for you to do it. Again, if you want to recognize on this graph here, you can see the concentrations right here, right? So cyclin E, it's starting in G1, right? And it's rising and it's rising, it's rising. When it has a large amount, woohoo, where do we get to? We move to S phase. That's what happens. And then it drops, it goes down because you don't need cyclin E anymore. But look, then you have a rising of cyclin A. When the cyclin A is really large, what do you have? You have drop into G2. And then after you reach G2, it drops. And then cyclin B is rising. When it's high, what do you move? Boom, you got into mitosis. And of course, when you get to the cyclin D, you back to G1. So this is what the graph is trying to show. So as long as you memorize this and repeating again to help you out. So let's put in red. From From mitosis to G1, you need cyclin D, right? Cyclin D to activate the enzyme. But remember, it has to be large amounts of cyclin D to move to G1. When this happens, then D, it goes back to being low. Once you want to go from G1 to S, you have large amounts of E. Once you have reached S, you drop the amount of E. When you go from ES to G2, you have to have large amounts of A. Once you get to G2, you drop the amount of A. When you want to go from G2 to M, you have to have large amount of B. And when you're going in your M and you're going towards G1s, you don't need B. So cyclins are rising and then dropping. They rise to activate and then they drop after they have activated. That's the regulation of the cell cycle. It depending on these 4 cyclins that will activate CDK to do the movement. Hope that helps. So you can see here some of the checkpoints we have. Now there's a checkpoint that happens here at G1. That's just to see if cell is ready to go, if the cell is too big. Then if it is too big, okay, then it's time to go to S. And you also have a checkpoint happening at G2 because they want to see, have the cell grown big enough? Do you have 2 copies of DNA? Do you have enough organelles? Yes, you do. Perfect. Now, there's also a checkpoint that happens in mitosis. It's just to see if you have split things equally, if there is chromosomes that are going the right direction. All of this stuff they check and then they go through the final stages of mitosis. So this is what the cycle is all about. If I wanted to draw here where the cyclins are located, we can always do the whole process again from here, but we can put it in much clearer thing, right? You can now see that for M to G is DAB. So you look for M, and if you're going from M to G, what do you need? D. And then if you have D, that makes more simple. Then here will be E, and then here will be A, and then here will be B. So remember DAB, and then DAB, again, starting from mitosis to G1. That's the hard part. So mitosis, G1, S, G2, and then mitosis. Start from mitosis, and you have no issues. Mitosis with the D going to the G. Maybe that way can help you a little bit more in memorizing this cycle. Again, showing you, just make sure you get this thing going on. So M with the D going to the G, G with the E going to the S, S with the A go to the G2, G2 with the B going to the M, and the cycle keeps going, making sure you have these 4 cyclins to do the work. Now that we have done, let's do some review of mitosis, just to make sure you're on the right set. So here you have the drawings. You can see that right here, it's G2 of interphase. It's not mitosis yet. So in here, you can't see clearly. Let me do with the, wait, let me take this out. Let me put a highlighter here. If you look at this, it's not clear yet. Look at the difference between this one and this one. Obviously, this right here, it's more clear, the chromosomes, and this one is not so clear. So obviously, this one is interphase still, and you can see the centrioles are still together. They're not moving to the opposite side. In prophase, you start to see the centrioles moving to the sides, and you clearly can see distinctly that the chromosomes have been condensed and they're visible. That's how you separate the prophase from G2 or the interphase. Now, prometaphase, this is something that is before metaphase. This is the moment you can see that we still consider this as prophase, by the way, in which you can see that the nuclear membrane is breaking down, and you can see that the chromosomes are starting to line up with the spindles that are coming out from the centrioles. Then you have right here metaphase. You can see now that all the chromosomes are lined up in the middle. That was the spindles that put them there. And at phase, you can see that they have been split and they're separated and they are going in the opposite sides. And then you get to the telophase, where you can see that the formation of the chromosome nuclear envelope is coming back, and eventually the chromosomes will become invisible again. So this is the 4 stages, and of course we have kind of put G2 and cytokinesis there, but these are not part of mitosis. Here's an example, interphase in the drawings. You can see it is clear here, and you can't see anything. So that's interphase. Here, you can see the 4-axis clearly. That means we're definitely in the visible mode. We're now in mitosis, specifically prophase. Why? Because you can see the spindles and the centrioles are starting to form. You can easily see that this is metaphase because they're right in the middle. Here you can see that's anaphase because you're splitting them. And then here, just this drawing right here, you can see that it's telophase because it's starting to split. And they're becoming invisible here, and the nuclear envelope is coming back. So these are drawings that you can also identify in a test if they ever show up. Here's another way to look at stage pictures like this. So now it's testing yourself. So if you look at this one, the first thing you can see is that they're in the middle. So obviously this will be what? This will be metaphase. Now looking at this one, now when you look at this, right? You look first that there is no spindle forming, and here inside, you can't see distinct visible chromosomes. So what would this be? This will be interphase because you don't see anything. However, on this one, look, now you're starting to see that they're becoming more visible, right? They're not like this one, and you can see that the spindles are starting to form, so this must be prophase. Usually an early prophase, but it's prophase. On this one, look, they're in the middle. Obviously, there must be mitosis. Ooh, which this means, this maybe I am wrong because look, they're separating. There's separation. They're not fully attached like this one. So you see how they're attached here? This one is actually anaphase. My mistake. And it's good. We learn to be mistakes, but then we get out of it. This is also most likely that we have all of them. Now, some students will think, oh, this is anaphase because they're going in separate ways. But look, this gives away that it's a telophase. You see the corners? So this is definitely a telophase. Now, this one right here, what would you say? Well, are they in the middle? They could be in the middle, but I think this is the late prophase because they're not 100% in the middle like this one right here. These are more organized. This one, definitely a telophase right here. This one, oh, you can see that they are starting to become visible, and that's a prophase for sure. And this is an anaphase. So you have to practice looking at these drawings as much as you can because sometimes, I wouldn't say sometimes, most of the times you will find one of the drawings of the mitosis that you have to know how to identify it. But here's the ugly part. That was just the drawings. Now, these are actually micrograph. These are coming from microscope. NIB can be such a pain. They will make you actually, they will actually put these kind of pictures in the IB exams. So you kind of get like a little bit tricky. It's like, okay, looking at this, it looks ugly, but it doesn't. It's almost the same thing. So where do we want to start? Let's look at this one right here. And the first thing you can see is this. Can you see different chromosomes being visible? Doubt it. So this for me would have been part of the interphase. We don't see much distinct happening. However, this you start to see some whites between them. You see these whites? That means the chromosomes are becoming visible. So this is more likely to be prophase. Now, some might say, well, why? I can't see centrioles. Yes, because the microscope cannot show you centriole. So it's hard to see a centriole in this situation. But look at the difference between, okay, let me highlight this so you can see what I'm talking about. Look here how it's still so dark. And here you have more white spots. Because there's more white spot, that's a prophase. Now let's look at the rest here. Look at this one. They all are lined up in the middle. And since they're lined up in the middle, most likely this is metaphase. Look at this. They're starting to be visible. And you can see a lot of white space, which is just like this one. So this probably is prophase. What else do we have here? Here, students will say, is this anaphase or is this metaphase? That's a very hard one to identify, very difficult. I still would say that this is metaphase. While this one, I would say that it's anaphase. However, they're not going to be that pain in the ass with you guys. They will give you a little bit more clear. But the splitting will be that you will see a little bit more white in the middle. This one, they still see in the middle. So I might be wrong, but don't worry. IB will make it a little bit more distinct. However, it's impossible to distinguish between prophase and interphase, because they're so close to each other, as you can see, this and that. So for example, this probably is interphase, this is prophase, prophase because you see gaps, prophase, prophase, prophase. The darker ones, like this ones, are the interphase. And this is all for the cell mitosis. That's a big video, my friend, but trust me, it does happen to fall a lot. Make sure you know it. Here are the questions that you can build up on it, as well as these. Go over them. Go over the video if you have to, and try to answer these questions. This is also found in the study guide. And see you later. Thank you.